Hey, it's your friendly neighborhood immunologist. And what I'm going to do today is start a series of lectures that I would do for a college class and present to you the brief history of immunology. So let's get started. Here we go. Oh, my head's in the way. I'll fix that. There we go. All right, so I'd like you to learn three things today. I'd like you to know the difference between variolation, vaccination, and passive immunity. And then know the difference between innate and adaptive immunity. That's really important, and that'll be a theme through a good number of the lectures. And then lastly, what are we doing right now? What are some cutting edge applications of immunology? Okay, immunology begins, what, 400 years ago in the 1600s. So around 1664 or 1684, depending on who you believe discovered cells. Then we move on to the earliest vaccination. Most people think that around 1796, the creation of the smallpox vaccine was the earliest start of immunology. Then we'll talk about passive immunity. That's actually being used still fairly often as a COVID treatment. So if you know anybody who went and got monoclonal antibodies, that's passive immunity. Anytime like a baby is breastfeeding, that's passive immunity too. All right, so immunohistochemistry, that is something I still do in lab pretty often, created around 1910. And then discovering the basis for transplanting tissue and cells and blood, that's called non-self discrimination, and that started in the 1950s. And then we actually didn't even know that we had long-term adaptive immune cells until the 1960s. Okay. Goodness gracious. So there were two inventors of the first microscope. There was Robert Hooke in 1664 and Anton van Leeuwenhoek in 1684. So Robert Hooke's microscope, I believe you can still go and see it on display in Washington, D.C. He was first, but Anton van Leeuwenhoek's microscope was more advanced. So most people just go ahead and mention both of them. So I'll tell you why, depending on who you ask, you might be glad that Robert Hooke invented the first microscope. So he actually published. You'll find out that the people who get credit for things published, because otherwise things get lost with time. So he published Micrographia, and he coined the term cell, because over here, he thought these little rectangles looked like monk's cells. So if we'd gone ahead with Anton van Leeuwenhoek's model, we would have actually called them wee animalcules instead of cells. But yes, if you would like to see the leather and gold tooled microscope he used to make these observations in his book, it's on display in the National Museum of Health and Medicine in Washington, D.C. All right. So Anton van Leeuwenhoek was a Dutch amateur microscope builder, and he did publish his letters, but they were published later in 1684. And yeah, he definitely thought they should be called wee animalcules, but that takes a long time to say, even though it's more fun. All right, so Hooke published first, and we call them cells. So now let's talk a little bit about the first attempts at creating immunity on purpose. So variolation is the earliest method of vaccination. And what that means is people took material, it's not pleasant, usually scab or pus from an infected person was collected and given to a healthy person. This had a variety of results. In China, it was pretty common to inhale dried scabs from someone recovering from smallpox. Sometimes that would activate the immune system properly. Sometimes that would give the person smallpox. It depends on how well the scabs were dried and heated. In Africa, sometimes people would take a pustule and rub it into, so a pustule from a person who's sick, and rub it into a scratch on a healthy person. The goal was to introduce whatever was going on in that infected person to a healthy person to give them some type of immunity. Okay. So things started to get a little bit more sophisticated in 1796. There was actually a farmer named Benjamin Jesty, and he noted that people with cowpox never contracted smallpox. We know why that's true now, because the cowpox virus and the smallpox virus are so similar that the immune system response, once trained with cowpox, fights off smallpox. All right, so he actually did take pustules from people, probably milkmaids at the time, and he took the pustules and he gave it to his wife and kids, and they were protected from smallpox. But he didn't publish. So Edward Jenner gets the credit, even though Benjamin Jesty probably did this first. Edward Jenner got the credit because he carried out the experiment systematically for over 25 years and he documented it. I'm not saying he's a hero by any means. Sometimes when you look back in history, you see some things that you're just like, wow, I can't believe you were allowed to do that. So what did Edward Jenner do? In 1796, he variolated a neighbor's boy. Do you mind if I borrow your son for the afternoon? He variolated a neighbor's boy against cowpox. And then he purposely exposed him to smallpox a dozen different times. But thankfully, the neighbor's boy was protected. And that's part of the findings that he published on. All right. So this isn't a vaccine just yet. So we're calling it variolation when you take a virus, a bacteria, whatever, in its native form, in its regular form on your body, and introducing it to somebody else. Like, it hasn't been manipulated, it hasn't been heated, it hasn't been purified, just straight up pus to a cut. Okay, so this is trending towards like a true vaccine. So Louis Pasteur actually invented the rabies virus about 100 years later, 1885. He administered the first vaccine for rabies virus to a young boy named Joseph. I'm not sure if it's Meister or Meester. And it's in this book. I have this book in my office. It is The Value of Believing in Yourself. I had it since I was little. So what did he do? He actually found rabies infected animals. He understood that the virus was actually in the spinal cord. So he dried and heated the spinal cord, thus manipulating the virus, destroying it. And then he injected it into this young boy. And he proved that vaccination had worked even after infection had occurred. So yes, often most of us don't ever get a rabies virus unless we have been exposed to potentially like a rabid dog or raccoon, something like that. All right, so now we keep getting closer and closer to the modern vaccine. Louis Pasteur, if you ever want to read about him, his career was insane. He like saved the French wine industry. He created multiple vaccines. Anyway, just an amazing scientist. So a few years later, no, this is before, 10 years before, he actually made a different vaccine. He made a cholera vaccine in chickens. And I've always been jealous of people who discovered things on accident and not to say that he wasn't an incredibly hardworking person, but he discovered the attenuated vaccine on accident. He prepared a vial of cholera and left it on the counter while he went on summer vacation. So he came back and he injected the chickens. And instead of dying, they got sick and then they got better. So here, what happened to the cholera was that some of it died while it was waiting on the shelf. Some of it was in a weakened state. So instead of getting the full-blown cholera, they had a weakened version of cholera and then recovered. So then he took a fresh batch of cholera and infected the chickens, the ones that had received the attenuated vaccine. And then the weakened bacteria conferred resistance to the full-strength cholera bacteria. So he actually also created the attenuated vaccine against cholera. Okay, so there is a few other concepts I want to go over. There's immune serum and passive immunity. So a few years after Louis Pasteur's discoveries, Emil von Behring and Shibasuru Kitasato found a serum that protected animals. Okay, so your blood has an incredible number of things floating around in it. And a lot of them come from your immune system. A lot of them also come from your liver. So immune components of blood protected animals from the same pathogen. All right, so active immunity is something you get from having a cold, fighting off the cold with your immune system. That's natural, that's active. You had a fever, you had muscle aches, you had chills. You can also have active immunity artificially through vaccination because they take a part of a virus or a whole inactivated virus, same for bacteria, and then they inject that into your body. Your immune system has a similar response, but typically less fever, less muscle aches, that type of thing. But you do still make an active immune response. Passive immunity is maternal antibodies. So in case you don't know this, the reason that a lot of doctors recommend breastfeeding for at least 6 months is because breast milk contains antibodies, and it's basically going straight from the milk into their mouth. It's going to like coat, their whole mucosal system and protect them from whatever the mom has antibodies for. So that's pretty cool. And there's also artificial. I brought this up earlier. If you know anybody who has gotten sick from COVID and they were asked to go ahead and receive monoclonal antibodies, that is passive immunity. Either they were made in a lab or somebody donated their blood that was purified to receive the monoclonal antibodies. Okay, so these two people, Emil von Behring and Shibasuru Kitasato, found that serum, so that's a blood component, that's the liquid part of the blood component, not the cells. The serum protected other animals from tetanus. And yeah, that's still how you can be treated for tetanus, by receiving antibodies against the tetanus toxin. Okay, so at this point, people understood that there were things in the environment, pathogens, where if you damaged the pathogens, gave it in a small amount, dried it out, you didn't receive the full infection. And then they realized that the liquid part of blood was protective. Why? They didn't really know. So Paul Ehrlich actually figured this out. If you've ever picked up an immunology textbook, the word antigen is all over the place. So what that means is antibody generator. That's what antigen means. It means it's typically a protein that creates an antibody generator, antigen. And what Paul Ehrlich discovered is that antibodies were very specific, And he came up with the lock and key theory, and that serum from one toxin did not protect against a different toxin. So toxin from bacteria A and toxin from bacteria B are different enough that one antibody wouldn't protect you from both. Okay, so let's see what happened when polio occurred in the United States. So this happened actually in the 20th century. It swept through towns in the summer months, which was unusual. And what happened is that children would lose the ability to walk. Children would even lose the ability to breathe. So polio actually means gray marrow inflammation. And that's an apt name for polio because it does affect the central nervous system and nerves. All right. So polio was isolated in 1909 by Carl Landsteiner and Erwin Popper. There were 2 monkeys that were injected with suspension from a 9-year-old boy who died from polio. And this is when they discovered that the spinal cords of the monkeys were damaged. And that makes sense because if children can't walk or can't breathe, all those signals are coming from the spinal cord. All right, so maybe most famously, FDR, Franklin Delano Roosevelt, was actually in a wheelchair because of polio. So in 1921, at the age of 39, he contracted polio. And he started the March of Dimes to help, let me move this, to help children who had been paralyzed due to polio. Okay, so here's a little girl over here receiving a polio vaccine. All right, the Salk and Sabin vaccine were created by 2 separate people in the 1950s. So there were about 50,000 new cases of polio every year, and Jonas Salk became a national hero when he got his polio vaccine approved in 1955. And then, just a few years later, Albert Sabin introduced an oral vaccine that ended up replacing Salk's. And at the time, I believe that the vaccine was also distributed in Russia. It was like a sign of good faith at the time. Well, because it's more important to put kids' health first. Okay, so there was an ethical dilemma with the polio vaccine because monkeys were actually used to create the vaccine. We didn't have quite the same laboratory capabilities as we do now. So what that means is more than 100,000 monkeys were killed in the course of developing the polio vaccine. Per one rhesus monkey, it supplied 65 doses of vaccine. So yes, it's not something that the scientific community wanted to keep doing. They wanted to give the vaccine to protect children, but then also spare the monkeys as well. So this is where cell culture comes from. Cell culture is using a cell that's been immortalized. And what that means is like a regular cell is taken out of a human or an animal, doesn't matter. And you can change some of their genes to grow forever. They actually take advantage of viruses that cause cancer. So these viruses go into the cells, change some of their genes, basically turn the growth genes on permanently. And that's how they become immortal. Because typically your cells only divide about 50 times before they're more or less genetically told to undergo self-destruct. All right, so John Enders and his colleagues, Thomas Weller and Frederick Robbins, were awarded the Nobel Prize in Physiology of Medicine in 1954. So this was a huge deal. They discovered that the polio virus could be grown in monkey kidney cells. So after that first monkey was sacrificed, they grew the cells indefinitely. And in fact, these cells are still being grown from that first monkey that they immortalized. So this saved thousands and thousands of monkeys. This technique saves many, many animal lives. So they're called immortalized, and this is the birth of cell culture. So this is from Janeway's Immunobiology. They're just showing what happened over time. So we've got 1940 here, 1990 here. So why don't we hear about polio? Because the two vaccines introduced were very effective. So the inactivated vaccine was introduced here, 1955, and there's already a decrease. And then Sabin's oral vaccine was introduced, and the cases are incredibly rare, less than 0 in 100 children. And the same is true for our other vaccine we talked about. Smallpox took a lot longer to eradicate, but the number of countries with one or more cases per month went from 30 all the way down to 0. And right before 1980, pardon me, smallpox was officially eradicated. Okay, so that's some history. Where are your immune cells? So they're all over your body because you could be infected at any part of your body. So the first part of your immune system is in here. It's in your face. You've got your lymph nodes here. You've got your tonsils and adenoids. Those are actually mostly B cells. People who don't have B cells, it's a very serious condition. They don't have tonsils because they're mostly made up of B cells. It's pretty wild. There's your thymus. That's where your T cells go to get educated. There's an entire lecture just on that. Your spleen filters dead or dying red blood cells through an immune cell called macrophages. We'll talk about those next time. You actually have lymph nodes in your gut too. That makes sense. You better check your food to make sure everything is okay. And there's lymph nodes in your groin, behind your knees, armpits. That's why light exercise and massage is actually quite good for the immune system. And there's tons of papers about biking, like cycling, and the immune system. It's very beneficial, particularly for your adaptive immune cells. So I tell my students the easiest thing is to just learn the adaptive immune cells, and then anything else is innate. Why not make it simple on yourself? So B cells and T cells are adaptive. They can last for years. They can last for decades by self-renewing. B cells make antibodies, those passive immune products found either created in laboratories, found in breast milk, found in donated blood. Those are all made by B cells. There's also T cells. I think the T is for telling other immune cells what to do. That's their job. They're like the general of the immune system. There's two different types. Again, a whole lecture on this, maybe even 2 lectures. So there's helper T cells. The CD4 is helper, and the CD8 is a killer T cell. Now, in the middle here, most textbooks don't talk about these, but if you get really into immunology, you might have run into these. Gamma delta T cells are thought to be like the original T cell. So the T cells we have here, they're thought to be the modern T cell, and gamma delta is thought to be the first T cell. So they don't recognize as many different types of pathogens, but they can react faster. There's also natural killer T cells, but I should tell you what a natural killer cell is first. So over here, we have some cells of the immune system that are full of these little dots. So you can see actually here, most of them are full of little dots. So let's talk about the smooth ones first. So you have your macrophage and your dendritic cell. They're sort of blobby shaped, and they're amazing at eating things. The process is called phagocytosis. So these two, their main job is to run around your body. Macrophages do a bit more running around than dendritic cells. They tend to sit in your skin, your tissue. And once they've acquired something through phagocytosis, they eat it, they break it down, and then they actually travel to find T cells. And when they find the T cells, they can activate them if they are a match. And we'll talk more about that. The rest of these have little dots. The dots are called granules. The granules are how they fight pathogens. So mast cells, their main pathogen to fight is worms. So this is a whole nother, I'm about to open a can of worms. So yes, the mast cell here fights like intracellular parasites and just all kinds of parasites. But because people in the US, people in Europe don't often encounter parasites, mast cells for us function in allergy. Yep, there's a lot to unpack there. There's even a doctor. Oh, I'll save it. No, I'll tell you a story. There's a doctor who had asthma and allergies, and he wanted to test his own hypothesis that introducing a parasite would dampen his allergies and asthma. And so he purposely infected himself with a parasite. And he believes that it did diminish his asthma and allergy and prove his hypothesis. It's called the hygiene hypothesis, but we'll talk about that more later. Natural killer cells were found to actually destroy cancer cells. They're one of our fastest and most natural ways to destroy cancer cells, although cancer, unfortunately, can adapt to avoid them. And basophils and eosinophils, they're also full of granules. They participate both in the removal of parasites and in allergy. And neutrophil, here you go. Immunology is a little bit gross, so I'll tell you, if you've ever popped a pimple, any whitehead you see, it is not bacteria, it's a neutrophil. So actually, a whitehead is probably neutrophils showing up, self-destructing. That's the white. That's like all the neutrophils that have exploded. They self-destruct on purpose. And by doing so, they spread the granules everywhere, destroying all the bacteria. So very short-lived, but they're effective. So if you ever popped a whitehead, it's actually you getting rid of neutrophils and the bacteria that they have probably taken care of. Okay, so here's a little chart summarizing some differences. Okay, so are they immediate? Adaptive or innate immune cells are. Adaptive immune cells take time, about 2 weeks. Recognizes pathogens generally? Yes. So for example, a macrophage can tell a bacteria apart from a virus. It will never remember that it did that, and it can't tell you what bacteria or what virus. So then recognizes pathogens specifically, that's a yes for the adaptive immune system. B cells and T cells here, they can tell you the tiniest differences between individual viruses and bacteria. Retains memory of pathogens. No for innate, yes for adaptive. And that makes a lot of sense because here, people who don't have B and T cells, they can get the same infection again and again and again, which is really unfortunate. But as long as you have functional B and T cells, they will retain the memory of that pathogen. All right, so in conclusion, variolation uses live infections to create long-term adaptive immune responses. Vaccination uses heat-killed or attenuated infection to create long-term immune responses. Cell culture allowed researchers to experiment and create vaccines without animals. The discovery of the antigen demonstrated that the immune response can be very specific. And now you know that B and T cells are adaptive immune cells that can create very tailored immune responses. So what are we doing right now? In 2016, actually, the first Ebola vaccine was ever created. There's huge treatment advances in cancer immunotherapy. people can actually have their dendritic cells removed from their body, trained to fight their cancer, and then activated. The technical term is proliferation. Now they proliferate and then they're put back into their body to fight the cancer. And then lastly, we now have treatments for arthritis and inflammatory bowel disease based off of antibodies. All right, that's it for lecture 1, and I'll see you next time.